Thank you, Mr. Chairman, for reading this quite long title of our work. So, in order to not replicate the title, I will just go through my presentation. So, the gram-negative bacteria like Mannheima hemolytica, Pasteurella multocida, or Histophilus somni may cause significant losses associated with respiratory tract infection, not only in beef cattle, but also in dairy cattle, from several weeks up to 6 months of life. So the objective of our study was evaluate the efficiency of the hyperimmune serum, which was produced against conserved antigens of the gram-negative bacteria, and therefore it has a broad interspecies cross-reactivity. So we would like to check if this serum might be used in prevention of BRD and also during treatment of bovine respiratory disease as a supporting treatment, not alone. So the wall structure of the gram-negative bacteria is slightly different from the gram-positive bacteria. There is an outer membrane, and in this membrane there are some proteins like outer membrane protein, like LPS, and in some cases also heat shot protein may be exposed. And these proteins are highly conserved, so they might be interesting candidates for subunit vaccines. So on this slide, this is the composition of our subunit vaccine. We use recombined heat shot protein 60, recombined outer membrane protein 40 from Histophilus somni, and also a combination of those proteins conjugated with the core oligosaccharide from Actinobacillus pleuropneumonia LPS, and everything was solubilized in the saline with the emulsigen as an adjuvant. So for production of so-called normal serum, we took the blood samples from 10 cows, and we pulled the blood, and then we produced the serum. And for production of the hyperimmune serum, we vaccinate with our subunit vaccine the cows, and this is the dates for the vaccination. And then in 2 last vaccination and 3 weeks after last vaccination, we collect the blood, we pull this blood, and from this blood we produced hyperimmune serum. So this is randomized control study performed on one big commercial dairy farm, which has kept the animals in 3 different places. This is the number of the heads of the cows in each place. And the protocol is when the calf is born, this was the Holstein Friesian red factor cows, so when the calf is born, is kept in individual hatches up to 6, sometimes 8 weeks of life, depending on the body weight. And then the calves are moved to the calf barn, which is located on a farm C. So our first experiment was to see if the passive immunization of the calves with the hyperimmune or normal serum will somehow prevent BRD. So we performed this trial in 2 periods. First for the calves born in the middle of the summer, so I will refer this as a summer study. And the second trial was for the calves born during the winter, so I will refer this as a winter study. But we follow up those calves born in those 2 periods, we follow up the calves up to 6 months of life. So we have 3 experimental groups. One group got the hyperimmune serum, the other the normal serum, and the control group didn't receive serum at all. The serum was given just after the calf was born, 100 of milliliters subcutaneously, and then at the moment when the calf was moved to the calf barn. We also measured the body weight of the calf, which shows the black dots in those dates. And also we made a clinical examination of the calf and take the blood samples, which is listed here in those dates. For the second experiment, we try to check if the hyperimmune serum will support treatment of the dairy cows during the BRD. So in order to divide the cows into the group, we use the calf scoring criteria, which was developed on the University of Wisconsin of Medicine. We slightly modified this scale by adding the lung auscultation. So together the calf could score maximum 17 points, and we divided the calf. If the calf scored 11 points or more, we assume that this is the severe BRD, and if the calf scored 10 points or less, it was, I will be referring to this as a moderate bovine respiratory disease. And in this experiment, the calf included to the study were between 3 weeks and 3 months of life, and we have two treatments. The one was the calves treated with the antibiotics and NSAIDs only, so this was like more conventional treatment of BRD, so this is the serum negative group, and this group was subdivided into the serum negative moderate BRD and serum negative severe BRD, and we have another group which included the calves treated conventionally, and additionally we added the hyperimmune serum, also divided into the subgroups. In each group there was 10 calves. So this time we gave the serum only once at the beginning of the treatment. We measured the body weights 14 days after and at 6 months of life. And we also perform clinical examination and take blood samples during the treatment and at 6 months of life. In samples taken from the calves, we evaluate the concentration of the IgG1, IgG2, and IgM. We also check the concentration of the acute phase protein, haptoglobin, serum amyloid A, and the fibrinogen, and we check if there was antibody seroconversion. So this is not typical seroconversion. We decided that if the results of the test from one blood sampling to another blood sampling will be increased by at least 2 pluses, then we counted it as a positive seroconversion. And it was against herpes virus, bovine respiratory virus, BVD, Mycoplasma bovis, and parainfluenza. As I already mentioned that we perform clinical examination of calves, and if the calf was sick, we take the nasal swabs, and we culture this in aerobic and microaerobic conditions. For statistical analysis, we use the factorial ANOVA, and the post-hoc test was the Tukey, and we also calculate the chi-square test to see if the distribution of the sick animals and healthy animals among the group is indifferent, and it depends on the treatment. And we also calculate the chi-square test to see if the percentage of animals above reference threshold for haptoglobin is different between groups. So this is the results for using passive immunization, so as a prevention, using serum as a prevention of the BRD. So for the majority of the laboratory parameters, unfortunately the results were not significantly different. We just noticed that for the IgG2, in some dates the concentration was significantly higher in the group which got the normal serum, and it was probably driven by the fact that those cows were more often sick. And for the acute phase protein, there was only significant difference in the first blood sampling, which might be due to the different amount of the colostrum which the calf drank. So the mean body weight was not significantly different between the groups. But when we counted how many calves were sick during the study, we noticed that in winter period, there was less calf show the respiratory signs in the group which got the hyperimmune serum. And then when we calculate the number of illness cases, so we calculate this by if the one calf was sick in 3 days, the date when we are collecting samples, we calculate it as a 3. So this is the total number of illness cases in all calves during all visits. So when we calculated it, we noticed that the group which got the hyperimmune serum in both trials was lower comparing to other groups. The table shows the results of seroconversion. So the highest percentage of the calves which seroconverted was in the control group. This is the number of the cases which shows the positive antibody for different pathogens. Unfortunately, the results from the bacterial culture wasn't so good. we have lots of contamination, but the major pathogen which was isolated in less than 20% was the Pasteurella. In the second experiment, so it was the use of the hyperimmune serum during the treatment additionally. Again, majority of the laboratory parameters were not significantly different. There was only the same as for the previous experiment, the difference in the beginning, but this time it was probably driven by the severity of the inflammatory process. The haptoglobin was not significantly different, and the immunoglobulin in the M class was significantly different in the last sampling, and this was due by the fact that those calves shows in the end of the study, again, signs of the respiratory signs. So it means that this serum definitely has no long-term effect. But what we notice that there is a difference in disappearance of the clinical symptoms. So at the beginning of the treatments, obviously all calves show the positive clinical symptoms. And then in the end of the study, there was the lower percentage of the calves which got the serum and showed at the beginning moderate respiratory signs. So in this calf, there was the fastest disappearance of the clinical symptoms, and this result was significantly different. The body weight at the 6 months of life was not significantly different, and the seroconversion this time was lower compared to the previous study, and none of the calf in this group seroconverted. We have isolation, the most often isolated pathogen was the Pasteurella multocida, and the isolation was from 30 to up to 50% depending on the group. So, in conclusion, the hyperimmune serum, but normal serum, decreases the calf morbidity, which shows the results of the first experiment. The hyperimmune serum in calves with moderate respiratory tract infection decreased significantly the duration of the clinical symptoms. Therefore, this new generation of the immune serum may be helpful in the future in limiting the use of the antibiotics in calves, in dairy calves. Thank you for the attention.